Through my own fault, because I wasn't here for 2 lectures and we've done too many quizzes, I would love to finish at the same place we usually finish, which means we're going to have to finish talking about metabolic regulation today, mostly sugar breakdown and synthesis. Not hard conceptually, we just need to kind of get through it. And then we're going to talk about the citric acid cycle, so hopefully you have that lecture too. And then next week we will move on from there. So then there's like 3 lectures left, and we're gonna plow through. Okay, so we're gonna get to integration of metabolism, which is a big deal and a cool story about long-term regulation of body fat, 2 cool hormones, leptin and ghrelin, and how your brain kind of integrates all this information about fuel source, metabolic needs, diet and exercise. That's a good place to end because it's a great summary of everything. You're going to interrupt me with the question already, Sean. Go ahead.
Quick question. Are you going to focus on this material, or are you going to
So there should be already on Sakai a study guide, chapter by chapter, of what you need to know, important concepts. I think it's been there all semester, so if you would have looked, you might have seen some of that. But that's specifically for the final. You don't look that far ahead. And, so that's content to focus on, what mechanisms you need to know, what equations you need to know. And then there's also a breakdown on what the final exam will cover. So I would say, so because there's the ACS standardized exam, and so my part only counts 80 points, I can't remember whether it's, a third is new material instead of a quarter. If we've had 3 exams and a final, if it was equally treated, it would be a quarter, so maybe it's a little bit more. But again, all of this, when we talk about regulation, you already know the enzymes of glycolysis, so it's not really new material, it's putting all the pieces together. So you'll see that the final is actually a lot of big picture kinds of things, compare and contrast protein structure with RNA structure, so having to bridge the whole semester. Not in a scary way, but hopefully in a useful way. Yes, Katie?
Are you going to send out the survey?
Yeah, I totally forgot. Can you email me and remind me? Somebody who has their phone, just send me an email. Just one. I don't need everyone to. And I will try to set that up tonight. I also have, and here we're not getting into the material we need to cover, I finished grading all your double helix essays, finally, and I've started on your rough drafts. I'm sure you're going to want to work on that all weekend. So for the handful of people that I've done that for, you're welcome to pick them up, and so you can come up and ask me, because it's not alphabetical, I just grabbed the top of the stack. I'm going to try my hardest to, by the end of the day tomorrow, get as many of those done as possible, and then over the weekend work on the ones that you submitted electronically and email them back to you. From what I've seen so far, you don't have to do a whole lot of work, so hopefully the most of the work is behind you, and now it's just fine-tuning. What I want you to do when you, again, we're not covering the material. What I want you to do is when you turn in your final paper, I want you to include your draft, so I know what I wrote on it, and write 2 sentences maybe about what you changed on your final paper. Okay, so if it's just, hey, you said it was great, I just found some typos, or, I realized that the end didn't really wrap things up, or it was too schmaltzy, or, whatever. And so think about your writing and how you've improved it, and whether writing a draft is a good idea or not. So just a couple sentences so that I can focus on how to see what you've changed. Okay, hopefully that makes sense. I haven't looked at the exams yet. You'd probably rather have me grade those, but probably grading the drafts is more important to you at this point. So I will try to get as much done as I can this weekend. Okay, so where we left off was regulation of the enzymes in glycolysis and gluconeogenesis that are the metabolically irreversible reactions that need to be tightly controlled. Okay? And so we finished glycolysis and gluconeogenesis. Now we're going to talk about the enzymes in sugar storage and release that also need to be regulated tightly because they're also opposing. So we have glycogen breakdown and glycogen synthesis. So it would make sense that you're not going to be doing both of those at the same time because otherwise you're just wasting the energy to make glycogen. So the 2 enzymes, so throughout the rest of the stuff today, the enzyme names that I want you to be familiar with are in red. So the important enzymes, and there's not that many, the important enzymes are in red, just to point out that they're opposite each other. Okay, so glycogen phosphorylase is the enzyme that breaks down glycogen polymer into glucose monomers. But remember, it doesn't just break them down into individual glucose monomers. We end up with glucose 1-phosphate. Okay, and that's going to get converted to glucose 6-phosphate for glycolysis. And then we're going to talk about what happens to glucose in the liver, which is different. Okay, so again, you don't need to know mechanisms for any of these. So we're essentially, I think, done with mechanisms. You'll see that we're going to glide through at a very superficial level, and I want you to appreciate all these pathways, understand the regulation. So for that, that's pretty detailed. But as far as enzyme mechanisms or drawing new structures, no, you don't need to do that. Okay, so glycogen phosphorylase is the enzyme that clips off a molecule of glucose from the non-reducing end of glycogen. And how does it do that? So if it used a water molecule, we would call it a hydrolase, but it's using an inorganic phosphate, so we're calling it a phosphorylase. Okay, so that should sound familiar. Where have you seen that action before? Wayback Machine. Glyceraldehyde 3-phosphate dehydrogenase uses an inorganic phosphate to clip off the covalent adduct. Okay, so again, glycogen phosphorylase uses a molecule of inorganic phosphate to make glucose 1-phosphate, which then can feed into glycolysis after it gets mutated. So again, this is just an overview because we know that glycogen is branched, so we can clip away from the non-reducing ends all of those glucoses as glucose 1-phosphate. What happens when we get to a branch? Okay, so there has to be another enzyme to deal with that. Again, it's not a big important deal. It's just to realize that if we only talk about one of the enzymes, we're not telling the whole story. So there is a debranching enzyme that will take off that branch point so that the phosphorylase can keep going. Okay, not a big deal. We just have to deal with that branch point. And then the enzyme that converts glucose 1-phosphate to glucose 6-phosphate is phosphoglucomutase. So again, shouldn't be surprising. It's a mutase. It just rearranges where that phosphate group is. So what is that glucose 1-phosphate, glucose 6-phosphate used for? Well, if we're in the muscle, it's a source of energy. Okay, so you have a little bit of glycogen in your muscles for rapid energy access. So if glycogen is broken down to glucose in your muscle, it's going to get used right away as a fuel source. However, where is most of our glycogen? In the liver. Okay, so we have, I don't know how many minutes worth of glycogen in our muscle and hours worth in our liver. So in the liver, we're probably not going to use that glucose as the starting material for glycolysis. We could, but we have other fuels that we can use in the liver for energy production. Any idea what those are? Just kind of looking ahead. So they're going to be fats. Okay, we could even use muscle if we wanted to, but in the liver, breaking down glycogen for glucose is not primarily to use it right away for energy. It's to release it into the bloodstream for the rest of the body. Okay, so if most of our glycogen is stored in the liver, we're going to break it down into glucose and then distribute it to all the other tissues that need it, including muscle. So once we use up the glycogen in our muscle. So once we've made glucose 6-phosphate out of glucose 1-phosphate, so here's our glucose 6-phosphate. So glycogen, glucose 1-phosphate to glucose 6-phosphate. So it turns out that the glucose, as it's migrating through the body in the bloodstream, isn't going to go through as glucose 6-phosphate, even though that would kind of make sense because then it's ready to use. And I think the problem is that there aren't transporters out of the cell for phosphorylated sugar molecules. I meant to look this up before I came to class, but that's my memory. And so we're going to transport the glucose out of the liver cell, out of the hepatocyte as free glucose. Okay, so we have to take the phosphate group off. So if that glucose 6-phosphate is not going to be used by a fuel source in the liver, used as a fuel source, it's going to go through one transporter into the lumen of the ER, into that kind of empty space in the endoplasmic reticulum. And now we have an enzyme, glucose 6-phosphate, that its active site, so it's embedded in the ER membrane, but its active site is facing the lumen. Okay, so it will dephosphorylate the glucose 6-phosphate only when that glucose 6-phosphate is in the lumen of the ER. And now we're going to transport the glucose out of the ER, kind of through the cytoplasm, and then out of the cell into the bloodstream. So we can also transport the inorganic phosphate out too. So if the purpose of, one of the purposes of the liver is to release glucose into the bloodstream for the rest of the body, why not have glucose 6-phosphatase, this enzyme that's going to clip off that phosphate group, why not have it in the cytoplasm? Well, why can't we just, why is that phosphatase not going to act in the cytoplasm of the liver? Yes. Right, so if hexokinase or glucokinase is in the liver cytoplasm, now if we're putting the phosphate group on and then there's an enzyme that will take it off, that's a futile cycle. So we don't want opposing enzymatic reactions to be happening in the same place. So this is another way of regulating enzyme activity by sequestration, which is a word that used to apply to putting enzymes in the nucleus or the ER lumen, and now talks about money in the government, right? So if we sequester the enzyme activity in a place where only the right substrate will go, then we're going to prevent dysfunctional activity somewhere else. So what other enzyme have we seen that with? Sequestering an enzyme. No. How about glucokinase? Remember, glucokinase hides out in the liver? In the nucleus of liver cells until glucose concentration rises enough, and then it comes out and can act. Does that sound familiar? So in that case, the enzyme is sequestered in the nucleus. Here, the enzyme, or at least the part of it that's going to function, is sequestered in the lumen of the ER. So we don't have a kinase and a phosphatase working in the same place at the same time. Yes, Liam?
That would be within the same cell, right? Like that
So
would be happening at the same time.
it's in the cell, but because it's in the lumen,
Yeah.
um,
I'm saying like the glucokinase.
The
that's
gluco-
in the same
yes, it's a liver cell
in the nucleus.
in the liver. Yep.
So, concentration rises.
Right, exactly. Yeah, so all this has to do with what's going to happen with that glucose. Okay, so glucokinase hiding out in the nucleus, what's the fate of that glucose maybe? So if glucose is going to get phosphorylated by glucokinase, is it going to go on to glycolysis? It's going to go on to glycogen storage. Okay, so that's a scenario where we have more glucose than we need for fuel. We're going to store it for later in the form of glycogen. So if we had, so again, that relates to these two enzymes too, because we wouldn't want to phosphorylate glucose to put it into glycogen at the same time that we're dephosphorylating glycogen to release glucose to the bloodstream. So we want to make sure that we're not, that the cell is not fighting itself. Does it kind of make sense? Yeah.
It has to pass the cytosol. How does the hexokinase?
Yeah, I think, maybe it depends where the ER is relative to the outside of the cell. Yeah, so this implies that there is some free glucose. I'm not sure. So is it going to get rephosphorylated and then come back in? I guess it could, but the hope is that the cell knows what it's doing. I'm not sure what the mechanism is for preventing that. Yep. Except, actually, so we do know that for glucokinase in the liver, remember the Km is very high. Okay, so that's sensitive to how much glucose is there. So it could be that glucokinase in the liver, even if it's not hiding out in the nucleus, it has such a high Km that most of the time it's going to be free of glucose, and so the glucose can leave the cell before it gets grabbed by the glucokinase. Okay, so that's breakdown of glycogen. So again, just a couple of enzymes to remember and general features about them. Now we're going to talk about making glycogen and the enzymes that are important for that. And so the steps are all on the left. Again, these are words. These are not reactions or mechanisms. So hopefully you can appreciate that. And so we're going to talk about each of these in sequence, but that's kind of just a reminder and a review slide. So the first step, so the funny thing is the building blocks for glycogen you would think are just glucose monomers. Okay, but as we've talked about before, sometimes to make a reaction happen, you have to activate one of the substrates, one way to activate the substrate is to use a molecule of ATP or its equivalent to make an activated form with either a phosphate group with a good leaving group or an adenylate, an AMP, which makes it a good leaving group or something like that. So we're going to see here that we actually start with a phosphorylated sugar. Okay, so what made that? Well, probably glucokinase, right? So if we have glucose, we have so much of it, we don't need it for fuel, we're going to store it for glycogen. Glucokinase will phosphorylate that and then pass it off to another enzyme, so NDP sugar pyrophosphorylase. Big mouthful, so you do not need to know the name of this enzyme. But what's going to happen then is it's going to use a molecule of some nucleotide triphosphate. ATP, in fact, we're going to see that it's typically UTP. Okay, and so we'll use that as our example. So now that glucose 6-phosphate is actually going to attack the alpha phosphate of UTP, releasing pyrophosphate, so the two phosphate groups together. So now we have sugar, 2 phosphates, ribose and base, so a sugar nucleotide or an NDP sugar. So one of the phosphate groups came along with the sugar in the first place. The other phosphate group came from the triphosphate. It was the alpha phosphate of that UTP. And so now now this is the building block for growing the glycogen chain. Okay, so the details aren't important, just that we have this NDP as the building block. Okay, and now the enzyme that is important is glycogen synthase. Okay, so it's not a synthetase. So this step of the enzyme doesn't use ATP, it's just a synthase, so it's making new bonds but not using ATP. And so so here it actually shows UDP glucose instead of just NDP. So if this is the glycogen chain that's already existing, so now this UDP is a good leaving group. Okay, so it's going to leave as that hydroxyl group on the 4-carbon, right, is going to attack at the anomeric carbon of that incoming glucose. Again, you don't need to know the mechanism, but just to say we have glycogen that now has one additional glucose molecule added, but the building block was not just the glucose by itself. Oddly enough, it wasn't just the glucose 6-phosphate. We had this other activation step. I don't know why that's necessary. You would think just having a glucose 6-phosphate would be enough to activate that molecule for nucleophilic attack. So whether that's a regulatory mechanism, I'm not sure I know. But that's what happens. Okay, and so if you notice, our glycogen chain had to be at least 4 units long already. So how do you ever get started? So here's one of the cool things that nobody ever tells you when you learn about glycogen, but now you're going to know. So we always say glycogen is a homopolymer of glucose. So it turns out at the core of glycogen, there's a protein. Okay, so in order to start making the glucose homopolymer that's glycogen, you start with a protein called glycogenin. And so the name of that hopefully kind of makes sense. So glycogenin, genesis, beginning, so the beginning of making glycogen. Okay, so it's a protein that has a tyrosine residue that acts as the first nucleophile to get the whole thing started. Okay, so the protein is both the catalyst and kind of the primer. So it primes the reaction, it gets it started. It serves as the first place where that sugar gets attached. So again, you don't need to know the mechanism. It still uses a UDP starting material, the building block, but now that first glucose is attached to the tyrosine residue on the glycogenin. Crazy, I know. Okay, so if you actually could see what glycogen looks like, there's actually a protein at the core that is kind of the scaffold that everything gets built onto, or at least that first chain, and then there's branching and branching on the branching, and then you end up with glycogen. Okay, so a little tidbit for you. So it makes sense that if we're going to break down glycogen and we're going to make glycogen, they better be regulated in unique ways. Okay, so we don't want them both to happen at the same time. So the two main enzymes, obviously glycogen phosphorylase and glycogen synthase. So those are the main ones that I want you to know about. And we're going to see how each of these enzymes, both of these enzymes are regulated in multiple ways. So hormones will regulate them, That kind of makes sense. You can probably guess which ones they are: allostery, phosphorylation, etc. So this is what we're going to talk about. So we've talked about phosphorylation as a regulatory mechanism. We've seen a few of those examples already, but now we're going to see another one. Okay, so remember, what is the phosphorylase that we're talking about here? So this is glycogen phosphorylase. And so when it's active, what does it do? It cleaves glycogen into glucose molecules, glucose 1-phosphate, right? So the active form of phosphorylase is called phosphorylase A. Really doesn't matter as long as you know that it's the active form. And so the active form of glycogen phosphorylase is phosphorylated. Okay, so we need an enzyme to do that. Or sorry, we need an enzyme. We need a kinase. So what's the kinase that does that? Well, it's called phosphorylase B kinase, because phosphorylase B is the inactive, unphosphorylated form. So we have an inactivated glycogen phosphorylase. A kinase comes in, puts a phosphate group on, and now that makes it active. And now that glycogen phosphorylase will cleave glycogen into the individual monomers. So what turns on that activity? So the enzymes that make, or the hormones that make sense, glucagon. So what does glucagon signal to the body? Low blood sugar. What might you want to happen downstream? Raise your blood sugar. How are you going to do that? You're going to clip off glucose from your glycogen. So if we need to activate glycogen phosphorylase to do that, we better have a signal that turns it on in response to low blood sugar. Okay, so glucagon is the hormone that says we need some more blood sugar. Glucagon will activate this kinase, which will then put a phosphate group onto the inactive version of glycogen phosphorylase, so that now we can clip glycogen into the individual pieces, release glucose into the bloodstream. What about epinephrine? What does that signal? You need energy. Fight or flight. Run away from the bear or fight the bear. Okay, so how are we going to get energy? We're going to break down a fuel that's kind of a rapid response fuel. Okay, so we're going to release glucose into the bloodstream so that we can do glycolysis, so we can get as much ATP as possible. So it makes sense that both glucagon and epinephrine would have the same downstream effect, right? Increased levels of AMP. Also, we need energy, so that's also going to activate. For every kinase, obviously there has to be a phosphatase to turn off the signal. Okay, so if the kinase puts the phosphate group on to phosphorylase, we're going to have a phosphatase to take it off and reset everything when we're not in the same, once our blood sugar rises or we're done fighting the bear and our adrenaline signal goes down. So now we're going to reset everything to an inactive state. Does that make sense? Okay. So, and I know this is a slide that you saw back in the signaling lecture, but hopefully now it makes more sense. Okay, both epinephrine and glucagon are going to have the same downstream effect of raising blood sugar, so we can either fight or our brain can work. What do we know about both of those receptors? So they're both G protein coupled receptors. So you knew that about epinephrine. Now you know it about glucagon in case you didn't already. So in fact, even though they're different hormones, they're going to produce the same second messenger, cyclic AMP, right? Which is going to activate cyclic AMP dependent protein kinase A. Again, you don't need to know the names of all these things, but just the downstream effect. And now that protein kinase A will activate the kinase, which will phosphorylate the phosphorylase, which will break down glycogen into glucose, which will raise your blood glucose. Okay, so this is what we call a signal cascade. So for every binding event on the outside of the cell, we make, supposedly, I don't know how they measure this, 20 cyclic AMPs, because each cyclic AMP, once it activates, protein kinase A can do that multiple times. So now you have an amplification of the signal. So it's not a 1-to-1, it's a 1 to 20, and then that 20 is 1 to whatever, so that it's a 10,000-fold amplification compared to what's on the outside of the cell. So it'll magnify the effect, and we can target more than one downstream effect. So if we have cyclic AMP, that can activate protein kinase A, which will then activate more than one target as well. Okay. So in the liver, glycogen phosphorylase. So again, if the active form is phosphorylated, we're definitely going to have to turn it off as well. Okay. So once we have high enough blood sugar, what's the signal that we need to shut everything off? Well, it would make sense if the signal is just available glucose. Okay. So glycogen phosphorylase, oddly enough, has an allosteric site that binds, glucose. Okay, so it turns out that once glucose levels rise, now there's enough glucose, there's a high enough Kd for this allosteric site, that the glucose will actually bind to glycogen phosphorylase. There's a conformational change, as always happens with allostery, and now that conformational change is the residue, I think it's a serine residue, that gets phosphorylated is now more exposed to the solvent, the cytoplasm, and now the phosphatase can come in and clip it off more easily. So glucose binding makes that phosphorylated side chain more accessible to the enzyme that will remove it. Plus, insulin will also activate that phosphatase. Okay, so both direct sensing of glucose by binding of the enzyme at an allosteric site and the hormonal effect of insulin equals high glucose will turn off glycogen phosphorylase, so we stop breaking down glycogen once we have enough glucose. Make sense? Yes. What?
So does glycogen phosphorylase not really bind glucose that well, or once the levels go really high, it's just unforced?
So presumably the Kd, so the dissociation constant, is high enough that at kind of basal levels where it's just being clipped off of glycogen and then secreted out of the cell, it doesn't see the glucose. So it doesn't bind until the levels build up because the Kd is high enough. Okay, so it's not particularly tight binding. There has to be a lot of it. Okay? Okay, so what that says is glucose levels will decrease glycogen phosphorylase activity because we're going to dephosphorylate that protein and turn it off, and it will actually increase glycogen synthase activity. So glucose is an important molecule, obviously, so both turning off glycogen phosphorylase and turning on glycogen synthase will happen. Okay, so how is glycogen synthase turned on? So glycogen synthase is active when it's dephosphorylated. Okay, so it's glycogen synthase is turned off by phosphorylation. Glycogen phosphorylase is turned on by phosphorylation. So those two enzymes that work in opposition with one another are regulated in opposite ways. Okay, so now glycogen synthase, which makes glycogen, is only active when there's not a phosphate group attached to it. Okay, and so taking off the phosphate group, so that's a phosphatase, happens in response to insulin, which makes sense. Glucose levels are high enough. So now we're going to turn on glycogen synthase so we can use up all the glucose that is built up. Glucagon, which signals low blood sugar. We're going to turn off that phosphatase so we keep glycogen synthase inactive. So we're not going to be making glycogen when we already have low blood sugar. That makes sense. Also, high levels of glucose will activate that phosphatase to turn on the activity of glycogen synthase. Does your brain hurt yet? Mine does. Okay, so all of these make sense. So we know that glycogen synthase and phosphorylase shouldn't be acting at the same time. It's just how are they regulated. Okay, so basically you know the hormones and what they indicate. You know what the downstream effect should be. It's just a matter of, okay, is it allostery? Is it phosphorylation? Does phosphorylation mean it's more active or less active? So that's kind of the details that it's important to get straight without being overwhelmed with. I know it's a little overwhelming. So do not panic at this slide. So we're going to make everything in the middle basically a black box, okay, that you don't need to know about. Okay, so you've already looked at the insulin receptor, right? So what kind of receptor is the insulin receptor? So it's a kinase. It's a receptor kinase. So when insulin binds on the outside, the insulin receptor substrate, which is just a protein, gets phosphorylated because the insulin receptor is a kinase, right? Now it kind of goes into the cell, does some things downstream, phosphorylates things. You don't need to know that part in the red circle. But the protein kinase B phosphorylates. So this is glycogen synthase kinase. Okay, so when the glycogen synthase kinase is phosphorylated, it's inactive, right? So in response to insulin. Otherwise, glycogen synthase kinase will phosphorylate glycogen synthase to turn it off. Okay, so when glucose is high enough that we're getting an insulin signal, now we're going to turn off the phosphate, the kinase, so that our synthase stays active in its dephosphorylated state. So insulin binding equals glycogen synthase active to store the excess glucose as glycogen. Make sense? Trust me. Okay, so obviously we want glycogen breakdown maybe to be coupled with glycolysis. So how does that happen? So we've already said that these processes are going to be different. The enzyme activities are going to be different in different tissues. So the muscles really just want to use glucose as a fuel source, right? The liver, however, could use glucose as a fuel source, but a lot of what it does is it supplies glucose to the rest of the body, right? And so muscle cells, we have to coordinate regulation. And in liver cells, we're going to have to think about how glycogen gets broken down, not necessarily for metabolism, not necessarily for energy, but to be released to the bloodstream for muscles. Okay, again, don't memorize this. This is only to help you categorize things. So we could have the same things happening in muscle and liver. Many times that happens. Okay, so glycogen can get broken down to make glucose 6-phosphate, but that's where it might diverge. Okay, so glucose 6-phosphate in the liver can deliver glucose to the rest of the body. In muscle, we're going to enter glycolysis, right? I guess that's what that little up arrow means, right? We're going to increase glycolysis, but in the liver, actually glycolysis is going to go down, and gluconeogenesis is going to go up. Okay, so in response to low blood sugar, we're going to feed everything to the rest of the body, either by breaking down glycogen to release glucose to the bloodstream, or make glucose from pyruvate. So different tissues have different responses. Okay, does that make sense? Don't memorize. Just use it as a global reminder. Okay, and so how does insulin do this? So insulin, again, the insulin receptor will phosphorylate things to bring glucose into the cell, so the GLUT4 receptor, not that you need to know the name of it. So then it will increase the activity of hexokinase in the muscle and increase the activity of glycogen synthase once levels rise enough that we have enough to store for later. What you probably have in your slides, if you printed them out, is I have a couple of summary slides that I'm not going to go through because it just is more peeling back more layers, but sometimes students find it helpful to categorize things. So especially the last two slides that are just a table that I made up. So what are the important enzymes in glycolysis, gluconeogenesis, glycogen synthesis, and glycogen breakdown, and how are they regulated? You know, so are they regulated by allostery? Are they regulated by phosphorylation? Are they regulated by sequestering? Okay, so just use that as a study guide. We're not going to go through it step by step. And there's a couple of summary slides from the textbook about what are the responses to low blood sugar, high blood sugar, epinephrine. Okay, so just use those as review tools. There's nothing new. It's just a way of summarizing things. Questions? Yes,
Did
Carter.
you just say that when glycolysis is up, that our glycogen synthesis in the liver is turned up as well?
So in the muscle. So sorry, this is muscle.
Right.
So if blood sugar is high enough, we will phosphorylate glucose, some of which could be used for glycolysis, but not necessarily. And once there's a surplus, so if energy needs are being met, then we could store it in the muscle for glycogen.
Okay, you said it two slides ago.
Oh.
The one after.
After that?
Yeah.
Oh. Going backwards?
Going forward.
Oh, forward.
Yeah.
Okay.
Here. So it's the muscle.
Mhm.
Glycolysis is happening. Is there gluconeogenesis happening in the liver?
There could be, yes. Yes. So you could have glycolysis happening in the muscle at the same time that you have gluconeogenesis happening in the liver. Yeah, yeah. Because, so again, this is all in response to low blood sugar. Okay, so the muscles are going to be taking in glucose maybe from the blood, as the liver produces it for the muscle, and using up whatever glycogen stores they have. So you could even see this maybe as a little bit sequential. So muscle will break down glycogen while it has it to do glycolysis, enter the citric acid cycle and everything else. And it also is going to take in glucose from the bloodstream. In the liver, we're going to break down glycogen to deliver it, plus we might be needing to do gluconeogenesis to produce even more glucose. Okay, if I said we're storing glucose, we're not storing glucose here. Yeah. Other questions? Okay, does anyone need to stretch? Okay, take a minute to stretch the beloved citric acid cycle. Okay, so we're going to jump back in. So pretend we just finished glycolysis. We ended with pyruvate, and now we're going to talk about what happens next. Okay, so we took a little diversion to talk about regulation and talk about sugar storage and breakdown, but now we're going to jump back in and pick up pyruvate at the end of glycolysis. Okay, there's Mr. Krebs. We have to pay homage to Mr. Krebs, Dr. Krebs probably. So the TCA cycle or the Krebs cycle, you've heard of all this. Don't need to know about that. And so so if we think about respiration, okay, so cells undergoing respiration, there's 3 stages, and they're all redox phases, okay? And so we can so we've gotten to here so far, so we've gotten to pyruvate. We're going to keep oxidizing our metabolites to feed into the citric acid cycle, so we're going to kind of talk about that part today. And then there's a citric acid cycle phase, and then there's the electron transfer in the mitochondria phase. Okay, so we're going to see how far we get into the citric acid cycle today, but ultimately all these play together in sugar metabolism. We're also going to see, of course, that other metabolites feed into the citric acid cycle, but we're going to go on our simplistic route of sugar metabolism. So where we started with glucose, we ended with pyruvate, now we're going to feed into acetyl-CoA in the citric acid cycle. So we're going to see the recurring theme. So if our fuels are getting oxidized, something has to get reduced. So we're going to see more cofactors being reduced. So we're transferring the electrons from our fuel into cofactors, which are then going to get harvested. So all the electrons of the reduced cofactors will get harvested in the electron transport chain in the mitochondria. Okay? So let me just take a quick poll. How many of you have done a lot of this in cell bio? Familiar with big picture? Okay, so there will be on our course evaluation a question about which courses you've had, cell bio, Chem 280. So every year we have debates over should cell bio be a strict prereq? Does it matter which one you take first? Does it hurt to not have cell bio first? Who cares about taking, there's overlap, but sometimes students find that the overlap is helpful instead of being boring. Maybe
it's really helpful.
Yeah.
Okay. Well, so what I usually tell students is whichever one you take second seems easier.
I'm testing cell signaling tomorrow, and
Awesome.
so that's going to be pretty easy.
Okay, good. Yeah, so whenever you see the material first, it seems a little daunting. So if you're taking it at the same time, you maybe have a few weeks of being overwhelmed, but they feed, you're studying basically the same material. So anyway, I don't want to make assumptions and rush through, nor do I want to bore you to death. Okay, so important molecule, I'm sure you've heard of it, acetyl-CoA. Okay, so what's the acetyl group? It's that, right? We're going to talk about what coenzyme A is in a minute, but first we have to get from pyruvate So friend pyruvate that you've learned the structure of, I know. So we have to get the acetyl part of pyruvate into acetyl-CoA. So we're going to lose CO2, so that's going to be a decarboxylation reaction. Plus, look at all that other messy stuff there. Okay, so I'm actually going to slow down, take a breath. So the pyruvate dehydrogenase complex is a very a very sophisticated enzyme machinery. Okay, we're going to do a couple of different enzyme reactions, even though it doesn't seem like a big deal. So you could say, well, why can't coenzyme A just kind of here pop off the CO2? It seems like it should be pretty obvious. So it's actually this big enzyme complex with 5 coenzymes. Okay, so the overall reaction that's going to happen is called oxidative decarboxylation. Okay, so pyruvate is going to get oxidized and the CO2 group is going to come off. Because pyruvate is getting oxidized, we're going to have something get reduced. And so we're going to see that it's actually a chain of redox reactions to ultimately result in production of NADH. So anytime we say dehydrogenase, probably your first thought is going to be NAD plus being reduced to NADH, and you would be correct. Okay, but the net reaction is an oxidative decarboxylation. We're going to see this again in the citric acid cycle. And in fact, we're going to see almost the identical full package of the dehydrogenase complex in the citric acid cycle. So even though it seems daunting now, in 15 minutes when we get to the next example of it, you can be like, oh, I know that one. Okay, so before we talk about the enzyme complex, so coenzyme A. So even though we always write CoA, sometimes we write CoA thiol to emphasize that it's a thiol, that is the messy yuck of coenzyme A. So now you see why we always just call it CoA, right? But what do you notice about it? It's not going to be an extra credit, so don't even waste your brain space. Never, ever. It'd take me so long to grade that thing because I don't know what the structure is. Okay, so what do you see about it? Okay, so it's an ADP, I guess we could say, if you want to be thorough. There you go. An ADP, so where might that ribose have come from? Pentose phosphate pathway, right? Okay, it's actually phosphorylated. And then we have this thing in the middle called a pantothenic acid. So if you look in the back of a box of cereal, you might see pantothene as a vitamin. Okay, so what did we say in the first day of class, or the second or third? So often vitamins are used to make coenzymes. That's why they're important in our diet. Okay, and then the important part down here, the business end, is the reactive thiol. So what else do you see about coenzyme A, maybe just instead of just the individual components? Is it a small molecule? It's huge. And not only just huge, would you call it long? Maybe like a flexible tether? Okay. So again, we're going to reach and grab a substrate for another reaction. Okay, so coenzyme A, you could also call a flexible tether. We're also going to see, okay, so where have we seen, so we've already seen TPP before, right? And this is lipoate. Okay, so lipoate, that's that guy there. So you've also probably seen lipoic acid or lipoate on ingredients in cereal. Okay, so another vitamin. Thymine, flavin, those are all vitamins that we need in our diet to make these coenzymes. So we're going to see that this dehydrogenase coenzyme complex is in the citric acid cycle, very important metabolic pathway, so that's one big reason why we need all these vitamins. Okay, so even just for this one enzyme reaction that doesn't seem like it should be that complicated, look at all the stuff we need. Okay, and so lipoate is kind of cool. Not only does it have a long tether, okay, so this is a lysine residue on an enzyme. Okay, so the coenzyme is not by itself, it's attached to a lysine residue on part of the enzyme complex. So you have this long tether, and it can become reduced. Okay, so this disulfide there in the ring can be reduced to be 2 thiols, and it can also be acetylated. Okay, just a little tidbit for you. We're not going to talk about that yet. So we've got coenzyme A, so that's one of the 5 coenzymes, and the other ones are NAD. I should probably write it like that, I guess. So TPP, what's that used for? Decarboxylation. So FAD, FADH2, redox cofactor, NAD plus, NADH, and lipoate. Crazy big group of cofactors. Okay, so again, what do we need the vitamins for? Making all these things. So pantethine, niacin, riboflavin, and thiamine, very important for breakfast. Okay, and so here is our dehydrogenase complex. So again, 5 separate polypeptide chains, 5 coenzymes. And the weird thing is about this complex is that there's multiple copies of each of the polypeptides. So not only is it a complex of subunits, it's a complex of multiple of each copy of subunits. So there's a lot of chemistry happening there, so we can do the same reaction multiple times and everything's kind of clustered together. I'm not exactly sure what the advantage of that is, but there's a whole lot of substrate being turned over to product in a very small space, all surrounding this complex. Okay, so why we need multiple copies of that is not entirely clear, but what is important is the idea of substrate channeling. So we've talked about that with the enzymes of glycolysis, how even though we learn one enzyme activity, often those enzymes are hanging out together, so the product of one enzyme reaction is fed right into the next enzyme as a substrate. Okay, same idea here. It's not even so much enzyme substrate channeling as enzyme passing, a handshake passing down the line. Okay, so if pyruvate comes in here, so again, there are, I said 5 subunits. There's 3 subunits, 5 cofactors. 3 enzyme subunits, E1, E2, E3, E just standing for enzymes. They have unique functions. There are other E1, E2, E3 enzyme complexes in the cell, one of which we're going to see in the citric acid cycle. When we have these other E1, E2, E3 complexes, E3 is always made from the same gene. E1 and E2 are separate genes, separate polypeptides, and assembled together. So it's kind of like this mix and match thing. So there's one gene for E3, it can combine with other E1 and E2 depending on what the substrate is for that reaction. So we'll see an example of that later. Okay, so if we start with E1, it makes sense that you'd need a separate gene for that to deal with pyruvate, and where the other E1, E2, E3 complexes don't deal with pyruvate, you would have a different polypeptide. So the dehydrogenase part of it will grab onto pyruvate. TPP, remember we learned, was a coenzyme that decarboxylates things. Okay, so now we have the released CO2 and the hydroxyethyl TPP, so a covalent adduct that again can get passed down to the next active site. And now what's the next active site? Well, it's on enzyme E2, and now we have this lipoic acid in the redox form. So the lipoic acid is going to grab onto the acetyl group. So now we have one thiol on the lipoate and one acetyl group. And now, again, flexible arm is going to move that one over to now the dehydrogenase. It's going to move that acetyl group over to meet up with coenzyme A, so that now we can release the product acetyl-CoA. So now we've released the product, but what do we still have to do? Reset everything. Okay, so that's one of the reasons why we need all these separate cofactors. Okay, we have to reset all the redox cofactors because now we've left. So we've left the lipoate as the thiol groups, and so we have to reset them back to the disulfide. Okay, so the dehydrogenase that's E3. So it kind of makes sense that you only need one gene for this in an organism, because if you're always resetting lipoate, it doesn't matter what the other substrates are. You can just kind of plunk that one on as an auxiliary factor. And so this E3 uses FAD. So now that FAD will oxidize the lipoate to the disulfide form as it gets reduced. But now we need to oxidize the FADH2 back to FAD, and to do that, we use a molecule of NAD plus to NADH. Crazy. Okay, so why do we need to combine those two steps, the FAD coenzyme and the NADH coenzyme? So I don't think we've talked about this yet, but enzymes that use FAD, FADH2 as a redox pair have that permanently attached. Okay, so FAD is not a soluble cofactor, but an embedded cofactor in an enzyme. So that's why if you look at those redox tables at the back of the exam, for any enzyme, any redox enzymes that uses FAD, the reduction potential will be unique to that enzyme. Because FAD is never by itself, is the point. FAD is always held onto by the enzyme that's going to use it. In contrast, NAD plus, NADH is a free cofactor. Okay, so it comes and goes, it can interact with multiple protein partners. So that's why if you look at the redox potential, it has a reduction half reaction potential that doesn't matter because it's always, it can work with another enzyme, but it's always going to have its own reduction potential. Does that make sense? So ultimately, the electron acceptor is NAD plus. So the electrons from oxidation of pyruvate, ultimately the net place that they go is into NAD plus. So the net reaction is pyruvate getting oxidized to acetyl-CoA with reduction of NAD plus to NADH. Make sense? And we're releasing CO2. So oxidative decarboxylation with release of the reduced NADH. Yes?
So is the reaction potential of FAD variable?
Yes. So depending on what enzyme it's with. Yep. Yes. In fact, when you look at the tables in the back of the book or on the exam, it will actually have the reduction potential for that enzyme, dot FAD or whatever. Okay. So now we're finally ready to enter the citric acid cycle. So another deep breath, right? So we've made the precursor, acetyl-CoA. That acetyl-CoA could also come from fatty acid oxidation. It can also come from metabolism of amino acids. So now, we can start from any fuel source and feed into the citric acid cycle. So how many reactions of the citric acid cycle are there? So there's 8 enzymes, right? We are going to look at these at a much more superficial level than glycolysis. Okay, so what do I want you to know? So I want you to know the names of the metabolites and the names of the enzymes, but not the structures. Okay, and we're not going to do any mechanisms, but I do want you to know features of the different enzymes. So, for example, is it an oxidative decarboxylation? Is it an oxidation reaction? Does it produce ATP or GTP, etc.? So just overview features. So we're going to go through each of these reactions like we did for glycolysis, but you just need to know it at a different level. Okay? So citrate synthase is the first reaction of the citric acid cycle, and it uses 2 substrates. So oxaloacetate, which you've seen before, right? So now we get to the real purpose of oxaloacetate, which is not just this intermediate thing in gluconeogenesis, but it's one of the substrates for the citric acid cycle. So oxaloacetate will basically grab the acetyl group of acetyl-CoA to release coenzyme A to do more of what it's going to do to make citrate. Okay, so citrate is the first product. Citrate synthase. So what do I mean by it's an ordered by substrate reaction? Okay, so remember when we talked about kinetics of enzymes, and if they had 2 substrates, they could either be ping-pong mechanism, right? Or they could make a ternary complex, and the ternary complex could be random or ordered. Okay, so we need a ternary complex for chemistry to happen, and one of those substrates has to bind first. Which one do you think has to bind first? Oxaloacetate. Did you peek ahead or you guess? Why does it make sense? Okay, so if we don't have enough oxaloacetate to keep the citric acid cycle going, then we're not going to occupy the acetyl-CoA. So this is the first cyclic reaction or cyclic pathway we've seen. And so it's important in cells to keep the citric acid cycle going. Okay, we're going to see that some of the metabolites of the citric acid cycle can be siphoned off from other purposes, but that's really a bad thing, actually. And so what we want is to always keep the wheel of the citric acid cycle turning, and so we don't want citrate synthase to hold on to acetyl-CoA and have to wait around to see if it's going to have a molecule of oxaloacetate. So it will only be activated to bind acetyl-CoA if there's enough oxaloacetate around, which means we're in a good metabolic place, we're continuing with the citric acid cycle, and we can keep making more energy. Okay, so here's the picture. Okay, so binding of oxaloacetate will trigger a conformational change, which will allow acetyl-CoA to bind. So acetyl-CoA can't bind on its own. Okay, next enzyme is aconitase. And so what kind of, do you have any guess as to what class of enzyme this might be? Lyase. Good. So whenever you can't quite tell, always guess lyase, right? Okay, so aconitase is a lyase. Really more for, so it looks like it should be an isomerase maybe, right? So it's actually named more for its mechanism than for the substrates and products. Okay, so we're going to remove a water molecule to make that double bond, and then we're going to add water across the double bond to make isocitrate. Okay, so an important feature of aconitase, those of you who have Dr. Dos Santos for lab maybe, or have talked to her otherwise, you know that she studies the synthesis of iron-sulfur clusters in proteins. That's her research area. And so aconitase has an iron-sulfur cluster in it. So it's an important thing to know. Okay, and so that iron-sulfur cluster is in the active site, and it helps to bind citrate. So it helps to bind one of the substrates. I told you we were just going to zip along today. Open your mouth because the fire hose is flowing, right? Which is not how I like to teach, I know, but we just, at this point, have to get through the important stuff. Okay, so the next enzyme, so we've kind of been setting the stage, but what do we really want to do? We want to keep making reduced cofactors because really the point of the citric acid cycle is to make those reduced cofactors that will then feed into the electron transport pathway to set up the proton gradient in the mitochondrial membrane to make ATP. Okay, so even though we get a little bit of ATP out of glycolysis, you're going to see we get a little bit of ATP out of the citric acid cycle. Most of the ATP that we're going to make by oxidizing fuels comes from kind of parceling away the electrons in reduced cofactors and then gaining that back later in the electron transport pathway. Okay, so this is our first true oxidative decarboxylation reaction of the citric acid cycle. So pyruvate dehydrogenase complex was really before the citric acid cycle. And so again, we're going to release CO2 and make either NADH or NADPH depending on the organism and the enzyme isozyme. Okay, so again, mechanism we're not going to study. Might make sense to remember that it's a manganese, containing enzyme. So we haven't seen too many metalloenzymes. So what was one metalloenzyme that you know of? Zinc was important. Alcohol dehydrogenase, good. The other one we just saw on the previous slide, 2 slides ago, aconitase is a metalloenzyme, so it has an iron-sulfur cluster. And now isocitrate dehydrogenase uses manganese. Okay, so those are the metalloenzymes we know about. And the product is alpha-ketoglutarate. So again, you don't need to know the structure, unless you're a structural person, and that helps you remember what it's called. Okay, and so now we get to the big messy complex you've already seen before. So alpha-ketoglutarate dehydrogenase complex, we should probably say, is functionally identical to the pyruvate dehydrogenase complex. And in fact, the 3 subunits are the same gene, the same polypeptide. We're doing the same chemistry, but now with alpha-ketoglutarate instead of pyruvate. Yes, Evander?
From the previous slide,
Nope, 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 nope. Just substrates and products. Okay, so alpha ketoglutarate dehydrogenase, same chemistry as pyruvate dehydrogenase. So we're going to bring in coenzyme A again. We're going to go through the lipoate again, but ultimately the electrons are going to get transferred to NAD plus to release NADH. Same thing. So now we have another CoA, succinyl CoA. Again, we've released carbon dioxide again. So what we're going to talk about in a couple minutes is we started with, so how many carbons are on the acetyl group that enters as acetyl CoA? 2. How many carbons are in oxaloacetate? 4. Okay, so we're going to start by adding 2 and 4 and getting 6. And now we're gradually going to peel off those carbons in the form of CO2. Okay, so we saw one oxidative decarboxylation, now here's another. And so we should be down to 1, 2, 3, 4 carbons. Yep. So now we have succinyl CoA. We've released that other CO2. And so sometimes on nasty exams, they will ask you for things like, if the carbon 1 position of glucose is labeled, where does it end up? Where could it come off first in the citric acid cycle? Yeah, but that was much easier. That was much easier. That was one step. Yeah, and so you'll see that at some point we end up with, so I think it's the next one, we end up with a symmetric molecule, and so you don't necessarily know, so that's where you get lost and it becomes ambiguous. Okay, I'm not going to ask you that, don't worry. Okay, so now we've made succinyl CoA, very similar to acetyl CoA, except now we have a 4-carbon attached. Okay, and now succinyl CoA synthetase. So here's our first step at which we actually produce an equivalent of ATP. So either ATP or GTP, depending on the enzyme. Okay, and again, it's called synthetase. Normally we've seen synthetases that use a molecule of ATP or its equivalent to make chemistry happen. So this enzyme is actually named in the reverse sense. So instead of consuming a molecule of ATP or its equivalent, we're actually producing ATP or GTP. Okay, so we're basically releasing succinate from succinyl CoA and producing GTP or ATP. Okay, so again, there's the mechanism on the right. Not a big deal for us, unless you love mechanisms. Okay, but now we get succinate, which, as you can see, is a symmetric molecule. Okay, so from now on, you know, the thing could have flipped around and we kind of lost track of which carbon was number 1 and which was number 4, etc. Okay, and then succinate dehydrogenase. So again, another redox enzyme. This one is going to freak you out because it doesn't use NAD plus NADH. So it's a dehydrogenase that doesn't follow the rules. Sorry about that. Okay, so succinate dehydrogenase makes reduced FADH2. So to what extent do you think that slide is accurately representing what happens? So why should you be suspicious of that? They're attached. So that makes it look like FADH2 just leaves. Okay, so we'll see that succinate dehydrogenase is actually one of the enzymes that's in the electron transport chain. So succinate dehydrogenase is a membrane-bound protein. And so even though it's making FADH2, that FADH2 just can't be released to the solution. So in fact, it delivers the electrons directly into the electron transport chain to come back to FAD. Okay, so all this is going to happen in the mitochondria, and it's going to be embedded there, and so it's not like the FADH2 is going to leave and float away. Okay, and so another way of accounting, besides just counting the carbons, is every reduced FADH2 ultimately will give us about 1.5 ATPs. Every molecule for NADH gives us 2.5 ATPs. I know it sounds kind of weird that you can half an ATP, but trust me, it works. Okay, so succinate to fumarate. Again, we're still symmetric there. and we reduced FAD to FADH2. Yes?
Are all of the enzymes that have FAD going to have a delta G equal to 0? It's like
That's hard to say. I mean, so certainly it has to get recycled,
Right.
but does that automatically mean it's going to be near equilibrium? So I guess there's two things to think about. There's the chemistry of succinate to fumarate, and then there's the redox. Obviously they're coupled, but does succinate dehydrogenase go the other way? I mean, obviously if its delta G is near 0, that implies it does. But I don't know about all flavin proteins being near equilibrium. I'm not sure. Good question. Good question, no answer. Okay, so fumarase will make malate from fumarate. And even though it looks to our eyes, no, that's not true. Let me back up. So that fumarate we would call a trans molecule, right? So the carboxylate groups are on opposite sides of the double bond, so it's a trans fumarate. I guess we could come up with some kind of cis molecule that would be called something else. And so fumarase is specific for that trans isomer. Okay, it's going to go through a carb anion. You don't need to know that, but to make malate. So what kind of enzyme, what class would you call this? Probably a lyase. Yes, very good. Lyase.
Do all lyses have an ACE in them? Because like probably ACE.
No, ACE just means enzyme. But maybe if it's not isomerase or synthetase. I don't know. Yeah, but the ACE just means enzyme. Yep. You knew that. You knew that. It's getting late. I know. And then finally, we made it all the way through. We're going to get to oxaloacetate. So malate dehydrogenase. So our last redox reaction is going to oxidize malate to oxaloacetate and give us our last molecule of NADH. Okay. So what do you see about the delta G there? Crazy high. Okay, so under standard conditions, you would never expect this to happen. So what is it that drives that reaction from happening when it doesn't look like it's ever going to happen? Well, we don't want it to build up. So if you think about, so it's actually not going to build up. So we want to keep a steady but small amount of oxaloacetate. So remember the first enzyme in the citric acid cycle, so citrate synthase, nothing's going to happen until it binds oxaloacetate. And so basically then we wouldn't have much free oxaloacetate sitting around, right? So that's going to drive, so it's like Le Chatelier's principle. So citrate synthase will grab the oxaloacetate, which will allow malate dehydrogenase to keep going. Once citrate synthase has the oxaloacetate, then it can bind acetyl-CoA to keep the citric acid cycle flowing. Okay, so both making oxaloacetate and using oxaloacetate are kind of the regulatory part of the regulation of the citric acid cycle to make sure that everything points towards if we have enough fuel, we're going to keep doing the citric acid cycle. Does that kind of make sense? And I know that's plenty of information. Let's see. So that's just kind of the summary, okay, in a much cleaner form to help you study. So we can keep track of the carbons, where they come and where they go, and the reduced cofactors, and the carbon dioxide. Okay, so that might be the kind of thing you might want to reproduce to help you study. So keeping track of, and you might even want to write in the enzyme names there. So we only get one ATP equivalent. There's only one step that makes either ATP or GTP, but we have all these reduced cofactors, which we're going to see are going to feed in to give us more ATP in the electron transport pathway. Okay. Any questions? We will talk about regulation of the citric acid cycle, and then electron transport and lipid fatty acid metabolism on Tuesday. I will stick around if anyone has questions. I know we are out of time, but I can stick around.